We at the Crowther Lab focus on what is the role of the Earth system, the biological system on Earth, in understanding and mitigating climate change. And no small part of that is its microbiome and the fungi that live in these forests. And to begin, I'd just like to place you in a forest. And when you think about a forest, you probably think about trees, which is good. The definition of a forest is an ecosystem dominated by trees. And most of what we know about forests is based on things we can measure above ground. So scientists go out into forests and they count how many trees are in the forest per unit area. They might wrap tapes around them to measure how big they are or how tall they are. They can do that pretty easily too. We even use satellites from outer space to remotely sense aspects of forest canopies from space. And all of this makes sense. This is a good idea. You know, this is above ground in forests is where photosynthesis happens. This is how trees convert carbon dioxide, this potent greenhouse gas in the air, plus water into sugars that trees use to build leaves, to build tree trunks, to build all this stuff. This is how trees buffer our entire climate system against climate change. We know every year the global forest system absorbs and offsets about 30% of all our anthropogenic carbon dioxide emissions. So we emit all this CO2, 30% is not there because we made trees grow faster. However, we also know as biologists or just humans that you can't just eat sugars and carbohydrates. You need nutrients, you need minerals, and trees generally can't get those above ground. They have to get those from the soil. And to do that, trees build roots, and trees build a lot of roots. And many ecosystems, there can be more, just biomass, just mass of stuff below ground in root structures as above ground in stems and leaves. This is a picture of a forester using a tool called an air spade, and he's just blowing away this top layer of soil. And what he's quickly revealing is this massive and this overlapping network of roots. And it becomes clear super quickly that it's extremely likely that that tree and that tree are probably interacting via their below ground networks, via these root networks. But when you look at roots very closely, and I mean so closely that you'll need a microscope closely, you realize that they're not really just roots at all. They're fungi. Most trees on Earth are in symbiosis, or in partnership with fungi. This is a high-resolution image of one of the smallest root tips in a root system at the farthest outer edge. And this thin layer of orange and white stuff, that's a mycorrhizal fungus. And it's enveloping that root tip. And what's going on here is that tree and that microscopic fungus are trading. Plants are trading some of those sugars that they fixed from the atmosphere, from that carbon dioxide, in exchange for nutrients that these fungi are exceptionally good at acquiring from soil. And one reason they're so good at that is because they build these vast hyphal networks in soil. So you can think of hyphae as roots, but for fungi instead of trees. And a handful of soil can contain over 100 kilometers of these fungal threads running through it. But thinking about them as thread is actually still way, way too big. They're actually much finer than that. They're about as fine as the individual strands of a spider's web. And because they're so fine, they can really enhance a plant's access to often limiting soil resources. And so it shouldn't be surprising that many plants and many trees allocate big time to these mycorrhizal fungi. These are 2 pine seedlings. You can see these feeder roots coming down. And this white material is that mycorrhizal hyphae. And it's masked up so much that you don't need a microscope anymore. You can just see it. In this particular image, at this particular early life stage, there's almost as much photosynthate, that fixed carbon, supporting the fungus, as there is supporting plant tissue. But this is just 2 pine seedlings. And so now I want you to imagine an entire forest with trees that can be dozens of meters high and hundreds of years old. That's a lot of fungi. And that's a lot of fungi because these organisms are critical to how trees interact with their soil resource environment. Many tree species cannot establish in the wild at all if these symbiotic fungi are not present. Now there's one last thing I want you to notice about this image on the right, and that's that it sort of looks like these hyphal systems of these two pine seedlings, it sort of looks like they're touching. It actually sort of looks like they're connected, and it actually sort of looks like they might even be the same fungus. And it looks that way because it is that way. These fungi can actually connect trees, even of different species, and they can connect an entire forest. And it really forces you to rethink about what we think, even as biologists, what we think about ecological and biological systems. We often think about them competing, but these things are actually all webbed and networked together. And the more we look at these, what we call common mycorrhizal networks, the more we realize, the more evidence we find that trees are sharing massive amounts of carbon. This was a study done just 3 years ago and published in Science, done in Switzerland actually, where they tented entire forest trees, big ones. And they measured how much carbon that this tree was fixing went to this tree. And they found that the amounts were huge. And they had seen these in seedlings. We had seen this in seedlings, and everyone had been skeptical. But as we scale this up, and as we look at bigger and bigger scales, we find that this interaction is really crucial to these forest trees and how they work together or apart. And all of this is to say that these mycorrhizas, these symbioses, can connect forests. They can allow trees within a forest to cooperate. And that's truly amazing. However, that's not to say that all mycorrhizal mediated interactions between trees are positive. Far from it. I'm going to argue today that some interactions can also be strongly negative if we go up to even bigger scales. And I think there's actually evidence for that. Even in the earliest experiments that were done to show that these trees could share resources. And these were experiments done by Dr. Suzanne Simard in Western Canada. She's at UBC now. This was done during her PhD in 1997. It was published. And she was working in forest science in Western Canada. And she'd always been taught that trees compete strongly for light, for soil resources. But she also noticed, as a natural historian, that fir and birch trees often co-occurred. And it was her opinion that this happened all the time. It seemed like there was a positive correlation between their abundances, not a negative one, not a fighting relationship. And she hypothesized, and she knew that fir and birch formed a set of mycorrhizas that could be shared. She knew, based on a bunch of early molecular biology and microscope stuff, that the root tips on fir were colonized by fungi that also colonized the root tips of birch. And so she thought fir and birch might be sharing resources via a common mycorrhizal network. But she wanted to prove it beyond what had been done before. And so she did an experiment. And she planted triplicates of trees throughout the forest in Western Canada, in British Columbia. And she planted fir, birch, and cedar just like this. And she knew, again, these fir and birch shared a mycorrhizal connection. And she planted cedar because she knew cedar also formed mycorrhizas, but of an extremely different type. And so for a lot of biological reasons, she didn't think the cedar trees could plug into the fir-birch network. And then, remember, these trees are absorbing carbon dioxide all of the time. This is how they eat. It comes from the atmosphere. And so she placed a bag over that fir tree, and she injected into that bag basically radio-labeled, radioactive carbon dioxide gas at a concentration that's not toxic to humans, but high enough that if she ran a Geiger counter, which is basically just a thing to detect radio-labeled radioactive stuff, it would crackle and beep. And so she basically had a wand that once she injected in there, it would go click, 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 click. And that fir tree is going to do photosynthesis, and so it's going to absorb that carbon dioxide and take on that radio label. And she left that bag and that radio-labeled carbon dioxide in the forest for a few hours, essentially. And she did this, there's like 30, triplicates of these things throughout the forest. And she went back a few hours later and she waved her Geiger counter, this radio wand, up the birch tree, and it crackled. the birch tree was full of radio signal. It was clear that carbon that was injected into that bag somehow got to that birch sapling. And then she waved it up and down the cedar tree. And it was silent. And that is what made it strong evidence that there was a carbon transfer from this fir tree to that birch tree, and that the mechanism was probably a mycorrhizal network. Because if it was just diffusing through the soil or leaking out of that bag, it would have gotten to that cedar tree just as easily. Now, the interesting thing to me about this experiment is it's mostly talked about as, oh, this is proof of this sharing interaction. But it's also proof that this cedar and this mycorrhizal network doesn't have access to that other one. These mycorrhizal networks have names. So the fir and the birch form what we call ectomycorrhizal fungal symbioses. And the cedar tree forms arbuscular mycorrhizal symbioses. And I'm going to argue to you today that these are fundamentally different ways of being a fungus, different ways of being a tree, and different ways of being an entire forest. And to do that, though, I want to dig just a little bit deeper into the natural history and the ecology of these very different types of fungi. And so the arbuscular mycorrhizal fungi are this ancient ancestral form of mycorrhizal symbiosis. There's actually evidence when plants first moved from water to land, because they evolved in water first, they started evolving this symbiosis before they even evolved the roots. These actual little blue things in here, this is the stain of a root cell. So this is actually a very thin root. And those blue circles are what we call arbuscules. Those are the heart of the fungus. And then these are the hyphae extending out of the root cell. And this is certainly a multifunctional symbiosis. Hundreds of experiments have implicated these organisms in things like plant pathogen protection, protection from animals and insects that want to eat them. Also drought tolerance. But I'm going to focus on their role on how plants access soil resources, particularly these limiting nutrients like nitrogen. And in that context, trees that form these arbuscular mycorrhizal symbioses are more common in ecosystems with higher levels of soil nutrients and faster rates of nutrient cycling. Now, in contrast, we have that burr-firch strategy, that ectomycorrhizal fungi. Ecto means outside. They form this sheath around the root tip. They actually never penetrate the root cell. And from that space, they build these hyphal networks into the soil. And interesting about them is they've evolved independently or convergently over 50 different times. There's not a single evolutionary origin. Lots of fungi have evolved to play this strategy with trees. And again, this is also a multifunctional symbiosis. But in the context of how trees access nutrients in the soil, trees that form these ectomycorrhizal symbioses are more common in ecosystems with low levels of soil nutrients and slow rates of nutrient cycling. And this fast, slow dichotomy is super, super important for what the rest of my talk today, so I'm just going to say it again. We really believe that these arbuscular mycorrhizal trees are an adaptation. These fungi are an adaptation to high levels of soil nutrients, and these ectomycorrhizal fungi are adaptations to environments with really low levels of soil nutrients. And just to give you a flavor of what kind of trees and plants we're talking about, when we think about arbuscular mycorrhizal trees, you can think of broadleaf things like maples, but you can also think of coniferous things like cedars. The redwoods of western North America, those are arbuscular mycorrhizal trees, as well as every piece of grass you've ever seen. And much of the lowland neotropics, so the Amazon rainforest, is arbuscular mycorrhizal. If we think about ectomycorrhizal trees, we often think of coniferous tree species like pines, spruce, and fir. But you can also think of hardwood things like birch. And as well as the dipterocarp rainforests of Southeast Asia, these are predominantly ectomycorrhizal. And what's important to know is that most tree species form one of these symbioses or the other. It's actually extremely rare for a tree species to form both of these symbioses. And that's super important to know because it means we can know a little bit about these symbioses if we know about the trees and if we know which trees partner with which fungi. During my PhD, I got really interested, and still today, in this question. I had known that these things might be adaptations to higher low soil nutrients, but I got interested in what if these mycorrhizal fungi aren't just players? What if they aren't just passengers? What if they engineer their soil environments, manipulate them to create high and low soil nutrients? What if they're actually building environments that favor their own strategy at the expense of the other one? Because we know most tree species on Earth are either partnered with one of these types or the other. There are other mycorrhizal types, but they're extremely rare. And so to think about that, I'd ask you to imagine an ecosystem that sits on an intermediate position on that nutrient availability gradient. Such that it's equally likely that an herbuscular mycorrhizal or an ectomycorrhizal tree establishes in that forest. And so there's some soil nitrogen or soil nutrient supply. We don't even think about it as nitrogen. Let's say these ectomycorrhizal fungi can somehow, through some mechanism, draw down that soil nutrient supply. That might lead to more ectomycorrhizal trees recruiting into that forest relative to herbuscular mycorrhizal trees, which would feed forward and further draw down the soil nutrient supply and maybe even exclude that herbuscular mycorrhizal player in the forest, which would then continue to feed forward and lead to the recruitment of more ecto trees, which would further draw down the soil nutrient supply. And so this would be what we call positive feedback to the ectomycorrhizal dominated state. These ectomycorrhizal fungi have manipulated the soil environment to favor their own strategy at the expense of the alternative strategy. And we could imagine herbuscular mycorrhizal trees could do this as well. If they could speed up the nutrient cycle, that could just send the whole damn thing backwards. So I spent my PhD studying this feedback. And what we found is that this happens. And we did experiments in lots of different ecosystems, and we showed that ectomycorrhizal fungi really do slow down the nitrogen cycle. And then at the same time, luckily enough, a bunch of other groups were studying herbuscular mycorrhizal fungi, and they found that these herbuscular mycorrhizal fungi really do speed up the soil nitrogen cycle. And so in theory, that should lead to basically dynamics where these forests push each other away. You know, these molecular scale interactions, these microscopic fungi should affect and engineer the soil environment to favor their own strategy, to favor their own network. And all these positive things are going on at the expense of the alternative strategy, in theory. But we can't do these experiments all over the world. To do the experiments we've done and other groups have done requires tremendous amounts of fine-scale work, excluding things, doing things very carefully, doing molecular biology and DNA sequencing. And we think that this should actually ripple up to affect the patterning of where forests are across landscapes, across continents, and across the Earth. And we wanted to know that answer. And so we took a very different approach from a very different vantage point. And this is work I've done in the past year in collaboration with the Crowther lab. And we started to ask this question. We basically went to this data set that the US Forest Service collects in North America. So basically what they do is there's foresters that go out and they monitor 100,000 plots of forests across North America. And in every single one, they identify every single tree species and they measure how big it is. And then they go back to that plot and they repeat that measurement every 5 years. And they've been doing this for decades. And through that process, they generate what we call demographic data. So we can look at basically tree birth rates and tree death rates. So birth rates, like we call that recruitment in forestry. That's just what is the rate at which new trees are entering the forest in the understory. And death rates are just the rate at which trees die. And we can use that to build models. So we do demographic modeling to understand how human populations are changing too. I'm sure you've heard about population demographics. It's often talked about in elections. For instance, you know, you might go out, and people do this, And they're like, okay, this age group has this birth rate, they have kids at this rate, and this age group has this birth rate, and this age group has this birth rate. Meanwhile, this age group has this death rate, this age group has this death rate, this age group has this death rate. And by doing that, and using those short-term rates that we can measure over 3 to 5 years, we can forecast how populations will change in humans. That's how we know we have a population bubble in the United States, for instance, because of the baby boomer generation after World War II. And we know we have a big healthcare debt coming up for us. But you can also split that up, those demographics. You can say, oh, this group of people has this birth rate, death rate, emigration rate, emigration rate. This group has this rate. And so you can start to model how like entire cities are growing or shrinking, countries are growing and shrinking, and how different populations within those countries are growing and shrinking. We can do that with forests too. And that's what we do. And because most tree species either form one of these symbioses or the other, we can actually use all this forestry information to see the forest not for the trees, but for the fungi. And to start asking, what are the demographics of these fundamentally different types of mycorrhizal networks, these ecto and herbuscular mycorrhizal networks. And so we did that. So here I'm plotting the probability of dying, an individual tree dying. And in blue, I'm going to be plotting herbuscular mycorrhizal trees, and in this green-yellow color, I'll be plotting ectomycorrhizal trees. And what I'm going to be considering is, what if that individual tree is surrounded by other trees? So in an herbuscular mycorrhizal forest, which is AM, ecto, EM. So this is the probability of an herbuscular mycorrhizal tree dying in a forest where all the other trees are also herbuscular mycorrhizal and has some probability of dying. And these are models we fit to control for the environment, all sorts of many complicated things about modeling trees. Ectomycorrhizal trees have a much higher probability of dying in a forest if they're surrounded by herbuscular mycorrhizal trees. And if that's true, that's positive feedback to the herbuscular mycorrhizal forest state. If they die faster than herbuscular mycorrhizal trees die, eventually that forest will just stay herbuscular mycorrhizal. And we can look at it in the ectomycorrhizal case too. Herbuscular mycorrhizal trees are much more likely to die in an ectomycorrhizal forest, holding everything else constant in the environment and soil conditions, much more likely to die than they are to die in their home environment when they're surrounded by herbuscular mycorrhizal trees, their own mycorrhizal network. And ectomycorrhizal trees are much less likely to die, and they're much more likely to die away. This is an example of a feedback. And so once a forest establishes one way or the other, it'll create death rates that favor its strategy over the other type. And this is something we've learned by looking at forest inventory data. We can also look at the birth rates. So this is recruitment data. These are new trees entering the forest population. And so there's some rate of new trees entering the forest population in herbuscular mycorrhizal forests, and it's much lower for ectomycorrhizal trees. And herbuscular tree recruitment, birth in ectomycorrhizal forests is low, and ectomycorrhizal birth is comparatively higher. And so we know that it looks like the magnitudes and the differences, the qualitative differences in these birth and death rates suggest that, yeah, these things should be pushing away from each other. And so the next thing we did is we built a demographic model just like we do for human populations. We initialized a ton of forests at basically 0 to 100% ectomycorrhizal trees. So if a forest is 100% ectomycorrhizal, it's 0% herbuscular mycorrhizal, and that's because most trees form either one of these symbioses or the other. They're not mixing. And so you could be 50-50, or you could be 100% herbuscular mycorrhizal, and we just start them at a flat distribution. And then we also include growth dynamics, because that's important for understanding forests. And then we let the simulation go. Let it go. Wait. No. There's a cool video. Where's the video? No! This is the worst day of my life. This is supposed to be a video. And if this video was playing, what you would see is that over 100 years, or 200 years, something, basically, these things would bobble around. And, you know, because there's stochasticity in this, like there's randomness in this. And when there's randomness in the environment, all this stuff matters. But eventually, it wouldn't look flat. It looks super peaked right here, and it looks super peaked right here at either all herbuscular or all ectomycorrhizal. You'd be much more likely to be an all herbuscular mycorrhizal forest where all the trees have the same fungal network, or an all ectomycorrhizal network where all the trees have the opposite mycorrhizal network, than in a forest where both of those mycorrhizal networks are present. They're pushing each other away. And that's the end of that simulation that was going to be so cool. And this is actually the observed data across North America. So this is just not a simulation. This is just, if you look at what it is today. And the point is, you can't get this distribution unless you account for these mycorrhizal types fighting. It seems to be more common to be ectomycorrhizal than herbuscular, but there's still two distinct frequency types. They're pushing each other away. These things have positive interactions when they're together within their same type. But across types, they seem to be engaged in this really strong fight. And you might really care about which type is where, because we've also linked which type is where to the ability of these forests to absorb and sequester carbon dioxide from the atmosphere. So again, trees are absorbing carbon dioxide through photosynthesis. That's how forests, the global forest systems, acts as a buffer against climate change. But trees don't live forever. Eventually they die, they fall on the ground, and that carbon then doesn't still go to the atmosphere. It can just stay in soil. Decomposer microbes have to eat it. It turns out those ectomycorrhizal fungi, when they slow down nutrient cycling, they slow down those microbial communities that eat dead stuff in general. And they build soil carbon. And we know this because I did an analysis of soil carbon storage in forests all over the world. And we controlled for climate, we controlled for all these other things that affect it. And we found that these ectomycorrhizal forests have 40 to 60% more carbon being locked up in soil compared to herbuscular mycorrhizal forests. And so it basically means that these ectoforests, if you hold everything constant, it's just the presence of these different fungal strategy, the slowing strategy rather than this accelerating strategy, leads to more carbon staying in the forest system rather than leaving it and going back to the atmosphere. And this has led other people to suggest that maybe we can manage forests for their mycorrhizal type, herbuscular versus ectomycorrhizal. And if you did that, you might be able to manage forests to offset climate change. Maybe we protect, or when we reforest systems, we plant ectomycorrhizal systems over herbuscular mycorrhizal systems because of the carbon sequestration benefit. And so maybe that could work. I'm worried about it though. But the first thing to know that is we need to get a map of where all these mycorrhizal forests are. And so the Crowther lab, we did that. We mapped, we used actually local scale forestry inventory data, and we scaled it with a lot of different things in global mapping to get a map of where ecto versus herbuscular forests are. So in red is ectomycorrhizal forests and blue is herbuscular. And they can be mixed if they're yellow. And so you see the high latitudes where it's cold are more ectomycorrhizal. Basically, temperature affects nutrient cycling too and favors ectomycorrhizal strategies. As it gets hotter, you go, you know, in temperate latitudes like North America, we have a huge mixture. And then in tropical latitudes, we have more herbuscular mycorrhizal types with notable exceptions, right? Like here is the Gilbertoni dendroni forests of Africa. These are ectomycorrhizal forests. Oh man, I keep blowing it. This is still working. All right. Maybe. Oh, yay. Southeast Asia, this is dipterocarp forest. These are ectomycorrhizal. And we actually, it's an open question right now. Why can these ectomycorrhizal forests stay in this hot place when there's this strong signature of environmental filtering? The leading explanation right now is it's actually these ecological interactions, these fighting interactions that are self-stabilizing, that are leading to the sustaining of those different forest types. But there's a lot of suggestions that we're losing those ectomycorrhizal forests. So this is an analysis I did of the relative abundance of ecto trees. So if it's 1, it's all ecto forests. If it's 0, it's all herbuscular forests. And the x-axis is nitrogen pollution. This is a type of pollution that we emit when we burn fossil fuels. We emit CO2 when we burn fossil fuels. We also emit NOx gas, which is nutrients, which is nitrogen, which is the thing that structures these forests. Also, when we do agriculture, essentially, we volatilize a lot of ammonia. And the rates at which we're inputting these nutrients to forest systems is an order of magnitude higher than what they ever were before. And we find when we map and we analyze where forest types are, where there's more nitrogen pollution, we've converted the forest from ecto to arbuscular mycorrhizal. And through that process, we're pretty sure we've also lost carbon from the soils, from the ecosystems. And that's just become CO2 that went back to the atmosphere. We're pretty sure this is an undocumented source of CO2 emissions. There's a positive feedback to climate change that we really haven't considered yet. And we think it might be big. We also know that fire suppression has shifted things from ecto to arbuscular for a lot of different reasons, as well as rising global temperature. Hotter soils cycle nutrients faster and shift things from ecto to arbuscular. There are feedbacks that allow them to resist that, but they can only resist so much. And so we're really worried that this is not really going to be a climate solution, but actually, a climate liability that we haven't really fully thought about yet. But I don't want to end just on Bummertown climate change, so I won't. But I hope one thing that you take away from this is that these forests are fungi. So when we think about a forest, we think about an ecosystem dominated by trees, and it certainly is. But it's also an ecosystem dominated by fungi. We know these trees cannot establish and exist in the wild without these symbiotic partners, and we know now that these symbiotic partners are critical to understanding the patterning of where different forests are in the landscape, and they're literally having wars across the landscape about who is where. These things are truly amazing, and I think, this is the forest microbiome. Just like we've created a whole disease in humans called dysbiosis. If the stomach microbiome is sick, you are sick. A forest can be sick because it doesn't have its microbes. And its microbes are predominantly fungi rather than bacteria. And so with that, I'd like to say thanks so much for taking the time to come out and learn about fungi. I don't do this work alone. I do it with a ton of people, and I'm lucky enough to convince a bunch of people that this is interesting and to support this work. And with that, I'd be happy to take any questions.